Thank you, Chair. And also thank you, Indian Academy of Science, for giving me the opportunity to present here. Today I'll talk about the molecular basis of chromosomal translocation. We just heard the talk about hematopoietic malignancies and other cancer. But here I'll talk a little bit about the molecular mechanism associated with the generation of translocation, particularly focusing on DNA conformation and the role of immune machinery. Before I start, I'll just give a brief introduction of kind of research what is being done in my lab, which are all centered around DNA double-strand breaks. It is one of the most lethal type of DNA damage normally occur in our cell. Now for the audience, if you are not familiar about a double-strand break, this is nothing but two neighboring breaks in both the strands of the DNA, as shown here, the close vicinity. Now, normally, whenever this kind of breaks occur in our genome, the continuity of the genome is affected. So therefore, there are no replication, transcription, and which can lead to many type of issues, including cancer, what we just heard. So, but whenever such break occurs in our normal cells, we have an efficient DNA break repair system, DNA double-strand break repair system, as shown here, which ensures the integrity of the genome, which means our cells are happy and we are happy. But in cases when misrepair of the DNA ends or the chromosomal end happens, and that is where chromosomal rearrangements and the translocations and such kind of abnormalities takes place, and it could lead to various diseases, including cancer. There are cases when there is no repair takes place in the nucleus, right? So that's what lead to cell death and then different mechanisms. And so my lab essentially work on all these aspects, but today I'll be focusing on more of chromosomal abnormalities associated with hematological cancers. So now when you look at the distribution of chromosomal translocation, mostly about 80–90% of leukemia and lymphoma are characterized by one or other type of chromosomal translocations. And going back to the history, somewhere in 1960s, that is the first time such translocations were discovered, just by using cytogenetics methods. Janet Rowley first described the Philadelphia chromosome, which is seen in chronic myelogenous leukemia. Then there were array of discoveries in next 20 years describing several translocations. And then early mid of 80s, that's where people started trying to look at why these chromosomes breaks and what is so special about it. So what they were trying to do was they were trying to clone and sequence, that's the era molecular biology started booming. And there were so many information generated in, say, next 10–15 years, sequencing several different translocation breakpoint junctions. Somewhere late 2000, that's where people like us started working on what is the meaning of all this information generated, why chromosomes break in certain people or certain genes, and then that gives some sort of selective advantage to those cells, which helps those cells to transform and then become cancerous later with an array of additional mutations. So just to again make it clear for non-biology audience, so these are two chromosomes, and whenever there is a translocation, essentially translocation is nothing but exchange of chromosomal arms. But the primary requisite for generation of such translocation is you need to have chromosomal break. Chromosomal break essentially means breakage in the genome, double-stranded DNA, right? And that's what is shown. This is a translocation between chromosome 14–18, which is normally seen in follicular lymphoma. Long time back, it's about, I think now it's about more than 20 years back, we have come up with an important finding where we showed that the break in this case, which is T14–18 translocation seen in follicular lymphoma, occur in BCL2. Of course, that was not our finding, but what we found out was there is something called major breakpoint region in BCL2, and it could fold into something called non-B DNA structures. I'm sure the audience here knows what is a bDNA, which is Watson-Crick hydrogen bonded double strand break DNA, right? So what we found out here was the DNA at this region, not in B form. We showed several evidence to believe that there is a single stranded character. And then we also went on to show that there is a protein called RAG, recombination activating gene. These are normally responsible for immune system diversification. I'm sure the students will know, right? And this protein can aberrantly cleave these kind of structures. That's what we showed during my postdoctoral studies. Then later when we started working here in IISc, that was one of the first questions asked, what is this non-bDNA structure we were talking about? Now, when you look at the literature, these are examples of several type of non-bDNA structures. So these are formed, for example, cruciform DNA, triplex DNA, hairpin, tetraplex, aDNA and zDNA. It's their textbook, well accepted one. And they all form when there are specific sequences, repeat sequences present in our genome. I'll not get into the details because time is limited. So essentially you need to have those specific sequence to have such special or secondary forms of DNA formed in our genome. And honestly, these structures were all described more or less same time, 53, 54, when original bDNA were discovered. But they never got enough importance, or honestly people did not believe that this will have some sort of biological function. Mridula, one of my first PhD students, showed that the structure formed during T14 translocation is something called G quadruplexes. These are the structures formed when there are stretches of guanines present in the genome. Then it can fold into some sort of a quadrat structure as shown here. And based on that, we come up with a model wherein we showed chromosome 18, that's where the BCL2 gene is present, break based on the DNA structure, but it's cleaved by RAG complex, which, as I said, is important for VDJ recombination during immunoglobulin diversification, and which is a sequence dependent process, whereas this is a DNA structure dependent process. And when we tried to look at several translocation junctions, information was available already in the database. We found out that not only in T14-18 translocation, BCL2 major breakpoint region, even other translocation breakpoint regions from the patients actually have these sequence motifs that can support formation of such secondary structures. And so that's where this is what one of the aspects today I want to maybe speak next 3, 4, 5 minutes, where we try to understand how BCR-ABL, these are the two genes broken during formation of Philadelphia chromosome. As I said, the one which was described in 60s, 70s. And then if you look at the patient breakpoint region, you see there are a lot of arrows here that indicate a particular patient breakpoint region. There are a lot of G quadruplex forming motifs, so which is again shown here. And what we did first was we tried to do a particular experiment, which is something called sodium bisulfate modification assay. I'm sure you would have heard about this name, sodium bisulfate modification, normally used for studying DNA methylation. So you unwind the DNA and then treat with the chemical, and the principle is nothing but sulfonation of cytidine when it is present in single-stranded DNA in acidic pH, and then so that the cytosine can get converted as a uridine. Right? And then following PCR and sequencing, you will read this as a C2T change, cytosine to thymine change, which is a easy way to find out originally in the genome whether the DNA region was single-stranded. So now what we found out was in BCR region there are many stretches of single-stranded area. And then we went back to the in vitro biochemical assay systems where we designed several oligomers from those G quadruplex forming motifs. As I said before, these are supported by stretches of guanines. So the complementary control region will be cytosines. Now, if you run on these kind of oligomeric DNA radio labeled on a native page, what you see here is, by the way, the structure formation here is dependent on potassium chloride. So in presence of potassium, you can see, I mean, one of the example I'll show you, the guanine started moving much faster compared to minus potassium gel, which gives an indication of, oh, there is something happening there. But then, of course, you do, mutation of each one of those structures to demonstrate that they are indeed something called intramolecular G quadruplexes. Then there is something, I'm sure you all heard about, circular dichroism, which can be used for studying structural variation of DNA and protein, right? So we could, I mean, again, I'm not going to get into those details. And then DMS or dimethyl sulfoxide protection assay can be used to, understand the base pairing in this structure. And this is a 2D model there. And now the issue here is, this is all oligos in vitro. People will ask, what is the relevance? It's all fine in a tube, but what is the evidence that it will form within the cells? But before getting into that, so here we cloned this region into a plasmid, which is like, double-stranded circular DNA. I'm sure non-biologists also will know that. This is a simple assay system, something called a primer extension. So denature and then radio-label the oligos. And then if there's a structure formed, G quadruplexes formed, then during the extension, you find a post site. Whereas in the complementary region of the DNA, where you will have cytosins, of course, no G quadruplex formation, then you don't find the similar kind of a pose. Now, if you mutate the G quadruplex forming motif, then you can see it's disappeared. And then if you change the positions of primers, you can also see the pose sites moving. So this is where we try to see within the cells whether this region can break. So what we did here is we used episomal constructs, and it's like a plasmid, but it can replicate. In this case, it can replicate in human cells, mammalian cells. Again, to make a long story short, we are able to replicate the translocation breakpoint region using this episomal DNA, and then this red sequence, what is shown is what is deleted, and these are all clones were sequenced, confirmed, and all those things. And then we went on to show that there is an enzyme called AID, activation-induced cytidine deaminase. This is a deaminase, and which can bind to these structure regions, as shown here, and then it can deaminate the cytosins and leading to the mismatch, and then finally, I'll show a model there. This was also replicated by using purified proteins. So this is our working model. So there are multiple G quadruplexes are formed at the BCR region, and then the AID deaminate the cytosins, and then there is something called AP endonucleases, which can cleave at the the mismatches, and failed base excision can lead to DNA break and translocations. And next 2-3 slides, I'll just summarize. This was another study where BCL6 breakpoint region seen in diffuse large B-cell lymphoma. Again, the patient breakpoint can form different types of structure, including cruciform DNA, triplex DNA, and then there are different clusters where even G quadruplex itself could lead to fragility. This was another example where we showed that RNA DNA hybrid formation or R-loop formation could result into, could make the MYC region fragile in Burkitt lymphoma. So to summarize this portion of the work, essentially our study shows that non-BDNA structures are formed at chromosomal translocation breakpoint region in lymphoid cancers. In presence of G quadruplex DNA, triplex DNA, cruciform DNA, R-loop structures contribute to the fragility at the breakpoint regions. DNA replication and transcription, then DNA supercoiling facilitate the formation of such structures within the genome. And enzymes like RAGs and AID can act on non-BDNA, leading to chromosomal breaks. Maybe another 3-4 minutes, I'll just try to talk a little bit about the this new property what we discovered about the RAG as well as AID. So this is what happens during normally during VDJ recombination in our B and D cells to ensure the immune diversity. So this enzyme, RAG1 and RAG2, it acts as a dimeric complex, binds specifically to recombination signal sequences. These are conserved sequences. That's why this process is also called site-specific recombination, because it just specifically cleaves up the 5' end of the heptamer. Then there is also a monomer in both these cases. So now what we did was we tried to do some biochemical assay system by using purified rags from human. This is the purified protein, and then we use the physiological substrate, which is the VDJ recombination facilitating substrate, and other one is the non-BDNA structure where bubbles are used. So Abni went on to show that when you incubate with the purified protein, actually even this case, when rags act as a structure-specific nucleus, there is a sequence specificity. For example, when cytosins are present, both top and bottom strand can be cleaved exactly at the double-strand, single-stranded DNA junctions. Whereas when thymines are present, it will cleave. The efficacy is much poor, but when purines are present, this protein cannot touch. Rupa went on to show that not only the sequence of the single-stranded region, even the flanking double-stranded sequence matters. And there are particular domains. There is something called a non-BDNA binding region in RAG1. If that is important for physiological function, whereas the ZN-C2 module contribute towards the structure-specific nucleus activity. Nishana, so she did a very interesting, simple experiment. What she did was she placed the monomer of VDJ recombination next to these kind of structures. Okay. So what she found out was now all these DNA structures which was otherwise not sensitive to RAG, become sensitive because you can understand because RAGs bind to the monomer, then cleaves at the single-stranded, double-stranded DNA junction. And she also showed even if you have a single nucleotide mismatch, that is good enough for good enough as long as there is a monomer is present. Now this was a cell paper published sometime in 2008 from my previous lab, where they found that CPGs are clustered near patient breakpoint region, and if there is a deamination of methylated cytosine, it could lead to TG mismatch, which causing the translocation breakpoint region. So at this point of time, we ask the question, how often such monomers, what we described before, may be present in the translocation breakpoint junctions in patient sequences. So we analyzed more than 3,000 patient sequences and found that about 93% of the cases you have monomers present at the patient breakpoint junctions, and of that 74% of the cases you have a CPG right next to that. So these are a lot of examples I will not get into. So now by using biochemical assays, we showed that purified rags can bind to those fragile regions. Not only bind, it can cleave. We also have done chip assays to demonstrate that rags indeed bind in those regions inside the cell. And this was LM-PCR or ligation-mediated PCR to show that these breakpoints seen within the cells are comparable to what is seen in the patients. Based on that, we come up with the model wherein rags bind to the mismatches created because of the deamination of CPGs, and then bind to the monomer and then cleave at the mismatch at the CPG site. This is where I am going to stop. Essentially, we found additional layer of regulation of rags in mammalian cells. We identified two microRNAs. One is MAR29C and other one is 29A. Both could regulate the expression of rags in a B cell, T cell stage dependent manner. I think this I'll skip. And to summarize this portion, RAG can act as a structure specific nucleus, cut non-B forms of DNA leading to chromosomal breaks. Additional mechanisms such as presence of cryptic signal sequence and CPG lead to translocation. Tight regulation of the formation of DNA structures by resolvases and expression of RAGs aid is important. MicroRNA 29C, 29A provide an additional layer of regulation during RAG expression. And this I did not show. RAGs are expressed in glioblastoma cells and could facilitate the formation of chromosomal rearrangement. This is something interesting because generally people believe this protein is expressed only in B and T cells, and we are working more on that. These are some of my collaborators, and then funding from all government agencies, and also some private agencies. I'm really thankful. And this is my current group. Then of course a lot of old members also contributed to the work what I presented today. Thank you.
Very interesting talk. I was just simple question. I mean, how frequent are these non-number-like sequences in normal humans?
Yeah. No, this is something reviewers made us to evaluate. When we did, it's about 3 to 4 fold more than, less than what we have seen in the patient breakpoint region. Yeah, so this is something we checked. Okay, thank you.
So,